Okay, well, hello everyone. Welcome to the James F. Crow Early Career Researcher Award Symposium. A few quick reminders. Each presenter will have a total of 15 minutes. Aim for 12 minutes with 3 minutes for questions. We'll monitor this session for code of conduct violations. The session is being recorded, and questions can be typed in the Q&A window at the bottom of the screen. It's my great honor today to introduce our 5 outstanding finalists for this prestigious award. But first, I'd like to take a few minutes to give you a peek and some understanding about the man who inspired the award. In 2016, the Genetic Society of America celebrated the centennial of the journal Genetics, 100 years of publishing top-notch research in genetics, a wonderful accomplishment. The same year, we celebrated the centennial of another genetics giant, James F. Crow, pictured here. Jim was born in January 1916. He obtained his bachelor's from Friends University in 1937. He earned his PhD at the University of Texas in 1941 and joined the faculty at Dartmouth that same year. 7 years later, Jim moved to the University of Wisconsin-Madison, where he spent the rest of his life. Jim died January 4th, 2012. Jim was a beloved teacher of genetics. He loved to teach, and his students loved him. Jim's lectures are still viewed as models of clarity here at UW-Madison. He inspired students to challenge themselves and was renowned for distilling difficult concepts, such as those featured in theoretical population genetics, down to their essence. Jim's enthusiasm for teaching boiled over into 2 highly successful textbooks. An Introduction to Population Genetics Theory, co-authored with his student, Moto Kimura, is still the gold standard in the field for theoretical population genetics. Jim's notes from his class, his undergraduate genetics class, were so useful to students that he decided to publish them in Genetics Notes, also known as Crow's Notes, for many years. Went through many editions and was translated into several languages. Jim was quite the teacher. Jim was also an inspiring mentor in genetics. This is a partial list of Jim's former graduate students and postdocs. I'll just give you a minute to read through them. What a who's who of population and evolutionary genetics. Jim often said his students were his legacy. Together with his students, Jim pioneered genetics research in a number of areas. He's perhaps best known for his theoretical advances. With Moto Kimura, he developed the infinite alleles model, a highly influential formulation for the field. He refined the concept of genetic load to measure how the average fitness of a population declines due to mutation and selection. His theory extended examinations of sex, inbreeding, meiotic drive, and other foundational topics in population genetics. Jim's lab was also a source of important empirical advances. His team discovered the segregation distorter loci in Drosophila melanogaster. His students discovered p-transposable elements. Jim also supervised the characterization of deleterious mutations occurring in nature using clever crossing designs. All of these discoveries have held up over time. They've been remarkable contributions to the field. Indeed, his characterization with his students of properties of deleterious mutations is still cited to this day. All in total, Jim published more than 250 papers, including many classics. I would say it's fair to postulate that it's impossible to imagine modern population genetics without the discoveries of Jim and his students. I was very fortunate to join the genetics department at UW-Madison in 2005, where my office was located next to Jim's. Imagine my delight in receiving that phone call. Jim was a wonderful colleague and friend. I miss him very much. The first 6 years of my appointment here were the last 6 years of Jim's life. And he shared many bits of wisdom with me, gave me wonderful advice, and was a treasured colleague. Today, I'd just like to share a few lessons, personal lessons I learned from Jim. Jim loved science, and he treated it as a social enterprise first and foremost. I think the only thing that Jim loved more than critically thinking about science and generating discoveries was to discuss science with other people. His office was a revolving door of not only luminaries, but people off the street. Indeed, Jim treated everyone with the same respect and admiration, coming to each conversation assuming that the person he was talking with would change his view. This was a wonderful characteristic of Jim's. Jim loved ideas, and he knew that they mattered. And so did their histories. Towards the end of his life, Jim sometimes lamented to me that the field of population genetics had been distracted in some cases by a focus on the latest technology and a crazy idea, he would say, that the newest technology would solve all of our problems. Jim preferred to toil with those long-standing issues that I would say mostly have still not been solved in population genetics. I think this was part of his attraction to theory. And he was hell-bent on understanding the origins of each of the important ideas in our field. With Bill Dove, he co-edited the Perspectives on Genetics in the Genetics Journal for many years. Indeed, Jim wrote 45 of these perspectives himself. Personal anecdotes threaded with scholarly accounts of the history of our field. Such a treasure. If you haven't read Perspectives on Genetics, I would strongly recommend that you do. Jim's commentary on the history of science, one that I remember, was upon discovering a bit of knowledge, he would often assume that someone had discovered it before. He would say, "It's new to me, but not new to science, most likely." Jim searched for truth, not credit. He was animated by an interest in understanding nature. rather than being acknowledged for his contributions. And just one example is this famous paper from Charlesworth et al., published in 1993, in which the seminal model of background selection was described. In the acknowledgements, you can see the authors are grateful to James Crow, who independently proposed a similar mechanism for helpful discussions. Jim once admitted to me that the Charlesworths gracefully offered to include him as a co-author based on the influence he had on their thinking. He simply responded, "The Charlesworths got it right. They didn't need my help, and I don't need their acknowledgement." Jim proposed a number of bold ideas during his career, and he never seemed to be afraid of being wrong. Rather than protecting his ego the way so many of us scientists do, he was once again searching for the truth. In fact, I watched over the years as Jim wrote personal letters, handwritten letters often, to people in the field who demonstrated that his thinking was incorrect, whether it was unpublished or published. He wrote these letters of gratitude. These were thank you notes. Jim once told me that he was more grateful to the people who showed him the error in his ways than the people who praised him. What a model citizen. What a model scientist. In 2016, the Genetic Society of America established the James F. Crow Early Career Researcher Award to honor Jim, to recognize Jim's enduring impact on students, and to encourage promising early career scientists. This year, we assembled a blue ribbon panel to choose the recipient of this prestigious award. It included Kelly Harris, Felicity Jones, Emily Josephs, Daniel Matude, Brandon Ogbuno, and Molly Schumer. I feel privileged to have chaired this panel with this outstanding group. We sifted through a large pile of really impressive applications and whittled it down to 5. These are the finalists you'll hear from today. I think you're in for a real treat. The applications of these finalists blew me away. Incredible. Each of them will give you a 12-minute talk describing their research, and I'm really looking forward to it. This is the randomly generated order in which they will speak, and in just a minute or two here, I will introduce Moi. Thank you very much. Okay. Welcome again to the Crow Award session. First up is Moises Exposito-Alonso. It's going to tell us about natural selection on the Arabidopsis thaliana genome in present and future climates.
I'll share my screen now. All right, hope you can see my screen. Hi, everybody. Hope you're also enjoying the virtual TAGC as I am. I want to begin by saying thank you to the James Crower Committee for the opportunity to share my work along with 4 other colleagues. What an honor. I'm really looking forward to their talks. So today I'm gonna talk about natural selection in the Arabidopsis genome. Ultimately, my group at Carnegie Institution for Science and Stanford University is interested in understanding evolution in response to climate change, which I like to think of as a new set of natural selection pressures that as evolutionary geneticists, we can study and begin making predictions about populations' likelihood to respond by adaptation or otherwise becoming extinct. But this requires a better understanding of the short-term natural selection over genetic variation of a species. So today I want to discuss briefly 3 questions. First, how strong is climate-driven natural selection at the genomic level? Is allelic selection variable across environments? And is natural selection additive or multiplicative? We use the Arabidopsis thaliana model as our system, which has 3 convenient advantages. The first is that it is an annual plant, so we can create experiments outdoor and count lifetime fitness because plants just stand there for us to seed offspring. Arabidopsis lives in extreme environments, so we expect that some genetic variation might be under selection in different environments, and we have over 1,000 genomes publicly available with seed stocks that we can use for experiments. So to answer the post question, we conducted common garden experiments with over 500 wild Arabidopsis strains from the 1001 genomes in 2 locations. One in Spain, which you see in this photo, that is at the warm edge of the geographic distribution of the species, and the other in Germany, which is currently the center of the geographic distribution, or at least in abundance. These experiments were done under foil tunnels, so we could replicate a Spanish rainfall and a German rainfall in half of the replicates as realistically as possible. And what this allowed us was to separate rainfall effects from location effects like temperature and photoperiod in terms of natural selection pressures. So here it is, the raw data that we generated, 25,000 fitness measurements, summarized in over 500 different genotypes in these histograms in different manipulated environments. Today, I'm gonna focus on 2 of those environments, Spain and low rainfall, and Germany and high rainfall, which are the natural rainfall in each location. And already from these distributions, we can see that natural selection in Spain must have been very strong in this artificial population, as many ecotypes or genotypes had fitness very close to 0. But I wanted to quantify this at the genetic level to understand what could be the potential response to natural selection in terms of allele frequency changes. So then I calculated marginally the association between allele states at a SNP in the genome for 1.3 million SNPs with the relative fitness, which is fitness divided by the average of the population, to calculate this association coefficient, which we call total selection coefficients, and capture both direct and linked selection on an allele. And in the case of self-fertilizing species like Arabidopsis, and this inbred panel allows us to calculate the expected change in frequency in a single generation after selection. So the results of this analysis done for all the SNPs in the genome have in the x-axis, represented in these Manhattan plots, where we have in the y-axis the significance that the S coefficient, the total selection coefficient, was different from 0. In red, we have those SNPs that passed a Bonferroni threshold, which in Spain and low rainfall were over 6,000. The top SNPs here changed as much as 25% in frequency in a single generation, and many SNPs just got fixed because many strains in this environment. So selection was likely polygenic, likely there were many causal variants in all 5 chromosomes that are dragging massive allele frequency changes in a single generation. In Germany, the architecture was likely also polygenic, although the signal is much weaker. We have still significant SNPs in all 5 chromosomes. And the maximum allele frequency change that we observed was 2.5%. So we've seen with these outdoor experiments that natural selection can be quantified at the genetic level, and a single generation is strong in the warm edge of the geographic distribution. But wasn't it in the same direction in both climates? To study this question, we have this graph where one dot is one allele in the genome. They're plotted by annual precipitation in the x-axis and seasonality in the y-axis of the geographic origin of the allele. I indicate in black the Spanish experimental environment precipitation, and color-coded in green are alleles that are favored in Spain, and in red, those that are not favored or that decrease in frequency. What we can see is that dots, that is alleles closer around the Spanish environment, were positively selected or increasing frequency. And the further away these alleles are, that is in increasing precipitation regions, the more negatively selected they become. This is a signature of local adaptation. Alleles that are found, or alleles are found in the environments where they have been selected in the past. And this selection must vary across space. So when natural selection varies across space, it's expected that allele frequencies between populations diverge. So we found that experimental natural selection measured in Spain paralleled an increase in FST per allele between geographic populations. So we have shown experimentally that selection is strong and opposite across climates, recapitulating local adaptation at the genomic level. To better understand the population responses to selection, it is key to understand the true underlying architecture of selection. So for the last part, I'm going to ask whether direct selection is additive or multiplicative. And this part is unpublished results, so I'm happy to get feedback. The relative distributions that we observed in our Arabidopsis experiments, those that we've discussed today and others, were highly skewed. And this was not only Arabidopsis thaliana, but also Drosophila melanogaster datasets and Saccharomyces cerevisiae datasets that have been published and we've reanalyzed. The most skewed distributions, which you might recognize, the one from Arabidopsis in Spain and low precipitation, were zero inflated and had a long tail. And even the most symmetric distributions seem to have some type of skewness. And this skewness is difficult to reconcile with an additive model of fitness effects. But the current tools for trait genome-wide associations are based on models. That is, they assume that as you increase the number of positive mutations in a genome, lifetime fitness is increasing additively or linearly. And this is typically justified because it's a good approximation in the infinitesimal model, and it works in practice. However, much population genetic theory posits that selection is multiplicative. So as you accumulate the number of positive mutations, your fitness increases faster and faster, and vice versa for deleterious mutations. And further theory, for example, involving the evolution of recombination or mutation load, has sometimes invoked other nonlinearities in the genotype fitness map architecture, such as synergistic effects that are more exponential than multiplicative, or diminishing returns effects that are kind of plateauing. So to understand the architecture of natural selection, with Rasmus, we developed a model for fitness genome-wide associations that can test additive and non-additive functions. We developed a zero-inflated normal likelihood in which each genotype is drawn from a true fitness. This true fitness is, for these genotypes, is either a sum of direct fitness effects of alleles or a multiplication. And in our model, we allow a power function where there's a hyperparameter that if differs from 0, it can create other nonlinear deviations. And we use likelihood optimization to fit the different hyperparameters, and typically with 1,000 SNPs. So here are the results. We saw the preliminary results. We saw that in all cases, Arabidopsis, Drosophila, and yeast, that we reanalyzed, selection was most likely multiplicative, but it's a complicated result. And I'll walk you through the example of Arabidopsis in Spain and low precipitation that we already know. So the likelihood was maximized under the multiplicative model, so AAC was minimized. However, under cross-validation, the multiplicative fitness model did not work so well. In the right, we have the predicted fitness from our model and experimental fitness of a fraction of the data that we hid from training. And what we see under the multiplicative is that there are many outliers. And what this produced was a lowering of accuracy in predictions. However, if we try to this multiplicative model using an additive model with a hyperparameter that is over 1, we get more conservative predictions in this extrapolation phase and get a higher accuracy. And we have confirmed these patterns with simulations under true multiplicative and true additive models. Okay, so to summarize, today I've shown you that natural selection is highly polygenic and very strong at the edge of the environment, the edge environment of the geographic distribution of Arabidopsis, that selection varies across climates and explains local adaptation, and that there is preliminary evidence that the architecture of selection is multiplicative. So in summary, we expect dramatic allele frequency changes in populations if there is genetic variation to climate selection pressures. Now we're interested in understanding how this is going to play out over multiple generations, as we also expect realized selection to fluctuate over time. So to do this, my group is conducting, in collaboration with many colleagues across the world, a globally distributed multi-year experiment where we are resequencing experimental populations in 45 locations using pulse sequencing to study allele frequency changes over time and across space. And with that, I want to acknowledge all the contributors of this work, my team, and all of you for your attention. And I'm happy to take questions now.
Hey, thank you, Moy.
I'll share my.
Oh. Okay. Can you see the questions, or you? I will read them. Here's one. Here's a question for you. Your results on strong selection in Spain seems to imply genetic diversity in Spain should be very low, since so many individuals die each generation. Is that consistent with the population diversity data? And this is a question from Kelly Harris.
That is a great question. So remember that our experiments were conducted with a very big panel of accessions, many of them coming from regions that are not Spain, obviously. And what we see is that essentially only locally adapted populations to this environment were surviving in our experiment. Now, in the German experiment, many more ecotypes survived that environment. So you would expect that diversity in the center of the distribution, that is in Germany, might be higher because all the ecotypes can survive. However, this does not parallel the genetic diversity that we observe in the species, because the origin of the species is closer to the current warm edge, so the Mediterranean. So the diversity that is present in northern latitudes is a subset that migrated in the last 10,000 years. So it does not parallel the current genetic diversity, to answer the question.
Great. Okay, one more quick question.
Yeah.
This comes from Wynn Meyer. Is the down and up pattern in association between selection p-value and FST significant? If so, do you have an explanation for the low FST alleles that appear to show strong selection in your experiment?
Yeah, great observation too. Yes, the correlation is significant, and is I cannot remember the value. It's in the paper, but a very good observation that some low FST alleles also had very strong selection. And my guess is that these alleles were rare alleles, so lower frequency alleles that didn't get to very high frequency in any region. So their FST is relatively low, but they were positively selected, or at least change in frequency in our experiment. So I think that's the reason.
Thank you very much, Moi. We need to move on to the next talk from Yuval Simons, Quantifying Selection on Heritable Variation in Human Complex Traits.
Yeah. Hi. First of all, thank you so much for choosing me, and I want to thank the organizers for organizing this wonderful online meeting so quickly. And as Brad said, I'm going to talk about quantifying selection on heritable variation in human complex traits. So from the onset of quantitative genetics, the nature and strength of selection on traits has been of central interest. Starting in Galton's 1877 a description of stabilizing selection and continuing with Fisher's seminal work from 1918, selection on complex traits has fascinated the founders of our field. In the latter half of the 20th century, much focus was given to the maintenance of genetic variation and the strength of selection acting on variance. Here you see some of my favorite papers about the subject, and they all make assumptions about the distribution of selection coefficients acting on complex trait variation. However, we now have a completely new source of data in genome-wide association studies, which provide a multitude of associations between genetic variance and various phenotypes. And for some traits like human height, we already have thousands of genome-wide association studies, genome-wide significant associations, and know that many of the variances identified in GWAS affect more than one trait. These results of GWAS provide us with a great opportunity to revisit these old models, update them, test them, and learn from them about selection. Today I will present our method to use the frequencies and effect sizes of genome-wide significant variance from GWAS and to infer the distribution of selection coefficients. First, I'll talk about our model. There are multiple lines of evidence suggesting many complex traits under stabilizing selection. So assume that the trait has an optimal trait value. There are and then individuals who have extreme trait values have diminished fitness, as symbolized here by the giraffes that are too tall or too short. But the vast majority of the population has phenotypes that are close to the optimum, these happy giraffes in the middle. And while the phenotypic distribution is concentrated around the optimum, each variant in the genome contributes to the variation and increases trait variation. Therefore, selection would act to limit trait variation because the wider the distribution is, the more likely we are to see individuals with extreme phenotypes and therefore lowered fitness. So each variant decreases the mean fitness because it increases the variation. Since selection acts to limit trait variation, it results in underdominant selection against trait variation. And the selection coefficient is proportional to the square of the effect size, since it's selection against trait variance. Once selection overcomes genetic drifts, it starts limiting allele frequencies, as you can see here in the site frequency spectrum. And where on the y-axis you see the density, on the x-axis the derived allele frequency, and we all use the site frequency spectrum, but we can ask the question of what happens if we flip this relationship around. If given an allele's frequency, what can we learn about selection coefficients? Let's look at an allele, suppose it's at a derived allele frequency of 0.5. For selection coefficients that are nearly neutral, the site frequency spectrum is identical for all of them because it's determined solely by genetic drift. So the likelihood of all of these selection coefficients is identical. Once selection starts overcoming genetic drift, the site frequency spectrum goes down, so the relative likelihood starts going down as a function of selection coefficient. Once selection is strong, we are very unlikely to see a variant in such high frequency, so the likelihood plummets. What would happen if we now looked at an allele at a different frequency? Suppose the allele was rare. Well, we now see the same behavior, but everything is shifted to the right, because now the site frequency spectrum only goes down for stronger selection. The overall result is that that knowing the allele frequency, we know that selection can't be much stronger than this limit, And this limit is set by 1 over the frequency. So frequency is set an upper bound for selection coefficients. Now we can ask a similar question about effect sizes. As we remember, the selection coefficient is proportional to the square of the effect size. With a single trait under selection, this is a simple linear deterministic relationship. But variance may affect multiple traits under selection. And because of that, and though the selection is still proportional to the square of the effect size on the trait we're looking at, since each variant also affects many other traits under selection, this is no longer a simple deterministic relationship. In our paper in 2018, we showed that this relationship can be approximated by a statistical model in which, conditional on the selection coefficient, effect sizes are normally distributed and the width of the distribution is set by selection. This means that for small selection coefficients, we would only see small effect sizes, but for larger selection coefficients in purple and red, we'll see larger effect sizes. And note that this model has only one free parameter that scales effect sizes in units of selection. Now, what happens if we know that an allele is at a certain effect size? So suppose an allele with this effect size. If selection is too small, we are very unlikely to see such a large effect size. We don't even see the likelihood yet. When selection becomes larger, this effect size becomes likely. And once selection is strong enough that this effect size is likely, the log likelihood almost plateaus, and it only slightly decays here because the distribution of effect sizes becomes flatter. What would happen if we looked at a different effect size? Suppose we looked at a smaller effect size. We see the exact same behavior, just everything is now shifted to the left. This is because we now, with a smaller effect size, you need a smaller selection coefficient to make this effect size likely. The overall result is now that effect sizes set a lower bound to the selection coefficient. And this makes perfect sense, since knowing that an allele is functional, it has an effect on the phenotype, we can deduce that it must be under some selection if we assume that the phenotype is under selection. We now can take these two things together and look at the likelihood as a function of both frequency and effect size. And we see that the likelihood of the selection coefficients is bounded. It's bounded from below, because if an allele has an effect, it must be under some selection. It's bounded from above because if allele has an appreciable frequency, it can't be under extremely strong selection. And our method of inference is simply summing these log likelihoods for all the genome-wide significant associations, that is the GWAS hits for a trait, to directly infer the distribution of selection coefficients at those GWAS hits. The reason we use GWAS hits is because that though each hit is probably not causal for the trait, it must be in tight LD with a causal SNP. So the distribution of frequencies and effect sizes for GWAS hits is a good proxy for the distribution of frequencies and effect sizes for the true causal SNPs. Note that in our likelihood method, we directly infer the distribution of selection coefficients at GWAS hits and our model's single free parameter, but we can also use it to indirectly infer the overall genetic architecture of a trait. We apply this method to various traits from the UK Biobank. For each trait, we ran GWAS on genotype variants from 300,000 white British individuals and infer the distribution of selection coefficients for the genome-wide significant hits, those with p-values less than 5×10 to the -8. Here you see the distribution of selection coefficients for staining in height. We see on the x-axis the log selection coefficient. On the y-axis, we see the density. And we see in grade a 95% confidence interval. As you can see, most hits come from selection coefficients around 10 to the -4 or 10 to the -3. So in these selection coefficients, you do have significant selection, but it's not much stronger than genetic drift. What does this result mean in terms of highest genetic architecture? I find it easiest to visualize in terms of the distribution of the phenotypic variance in traits. Here you see on the x-axis again the log selection coefficient, and on the y-axis you see the variance density. So if we look at current hits, they explain around 30% of the phenotypic variance of height, and as we mentioned, they are under moderate selection. This means that they are mostly common and they have moderate effect sizes. Though we only directly inferred the genetic architecture for GWAS hits, we can still infer the rest of the architecture if we divide these results for GWAS hits by GWAS power, that is the proportion of variance captured by hits at each selection coefficient. We can therefore infer that most variants under moderate selection are not genome-wide significant, and they account for an extra 25% of the phenotypic variance. These variants are mostly common, but have much smaller effect sizes than the GWAS hits. We can also confidently infer that strongly selected variants of low frequency and large effect sizes account for only a small fraction of the overall variation. The reason we have power to infer this is because we predict that with such large effect sizes, some variants should still be common enough to be identified in GWAS, and yet we see almost no low frequency large effect variants in GWAS. Lastly, we get to the nearly neutral range, where variants are common but have tiny effect sizes. Our predictions for this range are that, that such variants should have very tiny, because they have tiny effect sizes, are not and should not be genome-wide significant. Therefore, we have very large confidence intervals for our predictions for this range, and we cannot estimate the contribution of this range to heritability. Overall, the moderate and strongly selected ranges account for about 58% of phenotypic variation in height. They therefore account for the vast majority of height heritability. And the small amount of heritability that isn't explained by this range of selection coefficient probably comes from the nearly neutral range, where we are underpowered to estimate the contribution. We are currently working on a version of this inference, which will use not only genome-wide significant hits, but all SNPs across the genome when we take into account the LD structure, and will therefore be better powered in this region because it will use all SNPs. And we also applied a method to other UK biobank traits. Most traits have very similar architectures to height, albeit with a smaller number of variants and a smaller proportion of explained heritability. Here, for example, you see the inferred architecture for aged menarche, where again you see that most hits are under moderate selection. There is a sizable contribution from other moderately selected variants, but there is also only a limited contribution from strongly selected variants. Again, overall, we explain most of the SNP heritability, but may have a sizable contribution from the nearly neutral range, which we cannot yet estimate. The genetic architecture of BMI looks actually quite different. GWAS hits are still under moderate selection, but we infer that the contribution from other moderately selected variants vastly outweighs the contribution from GWAS hits. Furthermore, this contribution clearly skews towards the more neutral range of selection coefficients. This suggests that there might be quite a big contribution to BMI heritability from the nearly neutral range. However, because BMI has a much more neutral architecture, we have a much larger confidence intervals for our predictions for BMI. Let's have a quick recap. We showed that frequency and effect sizes provide upper and lower bounds on selection coefficients. We infer that the distribution of selection coefficients for GWAS hits for each trait. What we've shown is that for height and many other traits like aged menarche, the vast majority of the contribution comes from moderately selected variants. However, for BMI, the architecture seems to be dominated by nearly neutral variation. I would like to quickly thank my PhD advisor, Guy Sera, my current PI, Jonathan Pritchard, and the wonderful Hamanesh, Mustafa V, and Alisa Lynn Fortier, who helped me with the data analysis, all the founders, the organizers of this wonderful meeting, and you for listening. Thank you very much. I hope there's time for questions.
Thank you, Yuval. We have time for one question, and it's from Dmitry Petrov. What happens when variants are maintained by some form of balancing selection or local adaptation?
Yeah, so what we've shown in our theoretical analysis is that as long as local adaptation isn't very strong, we should be okay. However, for traits like skin pigmentation, our results, it's obvious that our model doesn't work very well because obviously there's been lots of local adaptation. And regarding balancing selection, it would indeed skew our results. So for example, for BMI, we have to exclude the FTO and the snippets and the FTO gene because presumably it's under a balancing selection maybe. And that's why it has a very, very large contribution to trait variation.
Okay, thanks very much, Yuval. It looks like we need to move on to the next speaker. Thank you.
Thank you.
Next up is Carl Veller, speaking to us about recombination, variance and genetic relatedness, and selection against introgressed DNA.
Okay, well, thank you, Professor Pessier. A big thank you to the organizers for salvaging this fantastic conference. And I'm very grateful to the selection committee for including me in this session. It's a big honor. I'll talk about the effect of recombination on selection against introgressed DNA. This is work in collaboration with Nate Edelman, Pavitra Muralidhar, who you'll hear from in a moment, and Martin Novak. It's motivated by our growing understanding that hybridization between separate species and the concomitant introgression of genes from one species gene pool to another's is extremely common. So this possibility became household news about 15 years ago with the report of Neanderthal DNA segregating in the genomes of non-African humans. But diverse genomic studies over the past few decades have shown that secondary contact, hybridization, and genetic introgression are just standard features of the histories of many, maybe most species. And the theoretical work that I'll present today is guided by three key observations that have emerged from genomic studies of introgression. The first is that introgressed DNA is typically deleterious to the recipient species. This can be for a variety of possible reasons, but importantly, the deleterious effect is typically spread across very many loci. So for example, it's been estimated that Neanderthal variants introgressed into humans were deleterious at approximately 1,000 loci, and comparable numbers have been estimated for other species as well. So because introgressed DNA is typically deleterious, it is eliminated over time by selection. And the second key observation that has emerged from simulation studies of this process is that the vast majority of this purging of introgressed DNA happens extremely quickly in the first few generations after hybridization. So this figure is adapted from a paper by Kelly Harris and Rasmus Nielsen on Neanderthal human introgression. And what their simulations revealed is that although Neanderthal ancestry has been removed by selection over the 2,000 generations since hybridization, in fact, the vast majority of that purging happened basically in the first 10 generations after hybridization. And to get a sense of just how extreme that effect is, I'll remove the log scaling of the time axis. You can see that that purging is really happening extremely quickly. The third observation I'd like to focus on is that when we look at the genomes of species with a history of introgression, we find that introgressed DNA is particularly concentrated in regions of the genome with very high model where introgressed DNA is weakly deleterious at many loci. And I'm going to try to characterize the role of recombination in the early purging of introgressed DNA and in setting up the positive correlation between the recombination rate and introgressed ancestry. To do so, it'll be useful to take a detour into a more classical problem in genetics, that is the variance in the amount of DNA that relatives share identically by descent, variance in their genetic relatedness. So for example, an individual has 4 grandparents and therefore receives on average 1/4 of its genome from each grandparent. But in fact, the amount that it inherits from a particular grandparent can vary depending on the randomness of the parental meiosis. And it's intuitively clear that the more that the parent shuffles its maternal and paternal DNA, the more even the inheritance from the offspring of grandmaternal and grandpaternal DNA. And so the variance across offspring should depend on the amount of genetic shuffling that's occurring. So we can be precise about this, and if we calculate that quantity in full generality, we find that the variance of genetic relatedness to a particular grandparent is given by this simple decreasing function of R bar. R bar is a metric of the aggregate recombination rate. It's defined as the average proportion of locus pairs that recombine per meiosis, and it's affected by a number of factors. So in species with many chromosomes, independent assortment of those chromosomes at meiosis contributes much more to R bar than crossovers do. The contribution of crossovers depends not just on their number, but also on their locations along chromosomes. So a crossover at the tip of a chromosome causes very little shuffling, but a crossover in the middle causes a lot. And R bar is also influenced by the spatial relations of crossovers with respect to each other. So for example, crossover interference, by spreading crossovers out evenly along chromosomes, tends to increase the amount of genetic shuffling that they cause. Okay, so what does this have to do with introgression? Well, let's look at the classic model of a pulse of introgression, where in this generation, some members of a foreign species with red DNA mate with individuals of the resident species to create hybrid offspring in the next generation. When those hybrid offspring have their own offspring, meiosis in them causes those second generation offspring to vary in the amount of introgressed DNA that they carry. And so if we ask how selection acts to purge introgressed DNA in these and later generations, we need to realize that the rate at which selection purges introgressed DNA is proportional to the fitness variance induced by introgression. And when introgressed alleles are deleterious at a large number of loci, that's just the variance across individuals in how much introgressed DNA they carry. That is, variance in introgressed ancestry. So where is this ancestry variance coming from? Let's zoom in on the second generation, and we can see that there's a contribution to ancestry variance from the fact that some individuals do not have introgressed ancestry and some do. But it's also the case that those that do have introgressed ancestry vary in how much. And so if we focus on that variance and realize that these individuals all have a grandparent from the foreign species, we see that the ancestry variance among these individuals is just the variance in genetic relatedness to a grandparent. And so because of this, the selection against introgressed DNA in that early generation is determined by aggregate genetic shuffling as quantified by R bar. So what effect does this have across species? Well, let's look at a high and a low R bar species. So humans have 22 autosomes, the independent assortment of which at meiosis leads to a very high value of R bar, close to its maximum of 1/2. Drosophila melanogaster, on the other hand, has only 2 major autosomes and no crossing over in males. And this causes a very low value of R bar, about 0.3. So if we simulate the purging of introgressed DNA, and these simulations are carried out in SLIM and calibrated according to parameters inferred for Neanderthal human introgression, we see this typical pattern where the rate of purging of introgressed DNA is very rapid in the first few generations and then starts to taper off. Those dots, incidentally, are analytical estimates from the calculations I've just showed you. If we now look at Drosophila with its lower value of R bar, we see that the rate at which it purges introgressed DNA in the early generations under the same model is substantially higher than humans. And the end result of this is that Drosophila purges an enormous amount more introgressed DNA than humans do. So while humans have retained about 50% of their introgressed DNA, Drosophila retains only about 7%. Or put differently, the amount of introgressed DNA that humans take 2,000 generations to eliminate, Drosophila eliminates in just 7 generations. So it's clear that the aggregate recombination process of Drosophila is a much more effective barrier to gene flow. And the intuition for this result is quite simple. Recombination is acting here to break up linkage relations among deleterious introgressed alleles. So in species with low value of R bar, those deleterious introgressed alleles are maintained in larger linkage blocks of more variable size, and that makes them easier for selection to eliminate. Importantly, these results hold across regions of the same genome as well. So regions of low region-specific recombination tend to purge introgressed alleles more efficiently. So here I've created a hypothetical species with 2 chromosomes, both of which receive the same number of crossovers per meiosis, but chromosome 1 tends to place those crossovers towards the tips, whereas chromosome 2 tends to have a more central distribution of crossovers. So chromosome 2 has a higher chromosome-specific value of R bar. And that means that chromosome 2 actually retains substantially more introgressed ancestry than chromosome 1. And you can see that that difference between the chromosomes is really driven by the early generations, showing how important they are once again. So that leads naturally to the third observation that I started with, the positive correlation observed across genomes between local recombination rate and introgressed ancestry. Now, everything I've spoken about so far has involved recombination's effect on linkage relations among deleterious introgressed alleles. And while those deleterious introgressed alleles are densely scattered throughout the genome, in fact, most introgressed ancestry is expected to be neutral or even sometimes adaptive. That means that the amount of introgressed ancestry retained by a species or by a genomic region is really the same thing as the amount of neutral introgressed ancestry retained, the amount of neutral introgressed ancestry that survives. For a neutral allele to survive, it needs to recombine away from its flanking deleterious alleles before one or both of those deleterious alleles are eliminated. And what I've shown you so far is that recombination affects the second component. That is, in regions of low recombination, deleterious alleles are eliminated more rapidly. But of course, in regions of low recombination, it's also the case that neutral introgressed alleles recombine more slowly away from their linked deleterious introgressed alleles. So basically, it's a double whammy for neutral alleles in these regions of low recombination. And so what we'd like to do is understand the relative contributions of these two components to the observed positive correlation between recombination rate and introgressed ancestry. That is, we want to know what's really driving this positive correlation. And to do that, we can decompose the observed covariance between introgressed ancestry and recombination rate across the genome into two components. The first is a component due to recombination's effect in unlinking neutral from deleterious alleles. And the second is a component due to recombination's effect on the rate of purging of the deleterious alleles themselves. And so if we look in our simulations, we see two things. First, as expected, we see positive correlations set up very rapidly between local recombination rate and introgressed ancestry. And we also see that the effect of recombination in unlinking neutral from deleterious alleles is a relatively small contributor to these observed correlations. Instead, they're mostly driven by the effect of recombination on the rate of purging of the deleterious alleles themselves. And the intuition for this result is that when deleterious introgressed alleles are sufficiently dense in the genome, most neutral introgressed alleles are a short recombination distance away from their flanking deleterious alleles. And so it takes them a long time to decouple from both of their flanking deleterious alleles. In the meantime, very early on, it's the case that recombination is affecting the rate at which deleterious alleles across the genome are eliminated. So what I've shown you today is that the rate of purging of introgressed DNA in these all-important early generations is determined by aggregate genetic shuffling as quantified by R bar. And so it's affected by things like the number of chromosomes, heterogeneity in the size of chromosomes, the number and location of crossovers. This means that species with low values of R bar should retain less of a signature of historical introgression than species with high values of R bar. This is also the case across regions in the genome. Regions with low region-specific R bar should retain less introgressed ancestry. I've also shown you that the positive correlation that has been observed in recent years between local recombination rate and introgressed ancestry is largely driven by the sort of subtle effect of recombination on the rate of purging of deleterious introgressed alleles, rather than the effect of recombination in decoupling neutral from deleterious introgressed alleles. And I'll finish with a more speculative point. So we've seen that recombination has an important influence on the fate of introgressed alleles. If introgression is as common as we think it is, perhaps it exerts a converse evolutionary force on the recombination rate. So for example, consider a modifier allele that reduces its local recombination rate. In doing so, it prevents deleterious introgressed alleles from recombining onto its background. So it maintains a fitter background and therefore increases in frequency. This is analogous to inversions being selected for in species with local adaptation. A more subtle question is what the fate of a modifier allele that decreases the global recombination rate is. And we know from molecular studies of meiosis that these global modifiers are expected to be very, very common. And here the logic is a bit more subtle. So a modifier allele that reduces the global aggregate recombination rate increases the variance in ancestry among its descendants. In doing so, it increases the fitness variance among its descendants, allowing selection to act more efficiently on its descendants. And so the allele ends up in fitter descendants and therefore increases in frequency. And so we see that quite generally, introgression selects for decreases in both local and global recombination rates. And so with that, I'll take questions, and thank you.
Hey, thanks, Carl. It looks like we're unfortunately out of time. You took the full 15 minutes. It was an engaging talk. Thank you very much. And you can see the questions. You can answer them offline here. Okay?
Yep, I'll do that.
Thank you very much. All right, we need to move on. The next talk is by Pavitra Muralidhar. She'll be telling us about predicted shifts in dominance, increasing the likelihood of soft selective sweeps.
Hi, everyone. First of all, thank you so much for inviting me to be part of this symposium, and thank you to TAGC for going virtual. Today, as Professor Pesur said, I'm going to be talking about predicted shifts in dominance, increasing the likelihood of soft. And this is done with Carl Veller, who you just heard, and Nate Edelman. All right, so dominance is a fundamental property of any organism with haploidy. And in this talk, I'm going to be focusing on diploids. And we can define dominance in many ways: dominance with respect to a particular gene product, dominance with respect to a particular phenotype, like Mendel's peas. But as population geneticists, we're primarily interested in dominance with respect to fitness, which describes how selection acts on the genotypes produced by the alleles segregating in a particular locus. Now, there are two fundamental aspects of dominance with respect to fitness that I will be drawing upon in this talk. The first is that gain-of-function mutations tend to be dominant and loss-of-function mutations tend to be recessive. And assuming that adaptation tends to occur through gain-of-function, this is often stated as beneficial mutations tend to be dominant and deleterious mutations tend to be recessive. Now, why this general pattern is the case was the subject of intense debate, in particular between Sewell Wright and Ari Fisher in the late 1920s and early 1930s. Fisher suggested that this pattern of dominant beneficial alleles and recessive deleterious alleles was due to the evolution of dominance modifiers, whereas Wright instead proposed a physiological theory, suggesting that metabolic pathways had some margin of safety, and so one loss of function or decrease of function allele would be unlikely to cause too much damage, but two might. Now, as it turns out, Wright's theory was mainly correct, and it was really primarily extended and elaborated on by Henrik Kasker and James Burns in the 1980s. And their work, which used metabolic control theory, has now been turned into the paradigmatic metabolic theory of dominance. And they were looking at the flux going through metabolic pathways and showed that it depends very little on enzyme production at any particular step in that pathway until that enzyme becomes limiting. What this means is that a single loss of function or a single decrease in function is unlikely to cause very strong deleterious effects, but two such copies would in fact cause deleterious effects. And so the heterozygote is likely to be far less affected than the homozygote. And since in the 40 years since that paper was published, there have of course been many experimental studies confirming this work and theoretical work extending it to new gene categories. Now, the second component of dominance with respect to fitness that I'm going to be referring to in this talk is that the dominance of an allele is not constant across selective environments. And this is sort of intuitive. Dominance with respect to fitness is defining how selection acts on three genotypes. So if selection is acting, say, in a different aspect of the phenotype in each of two environments, or is just acting differently in two environments, there's no reason dominance should remain constant. Now, with those two aspects in mind, it's interesting to think about the classical framework of selective sweeps, in which there's a deleterious allele segregating in the population, some environmental change occurs, and now that allele has a selective advantage instead. The usual assumption here is that selection is constant before and after the environmental change. However, we know that's not likely to be true, and in fact, we can predict the direction in which dominance should change. The deleterious allele is likely to be recessive with respect to fitness, while the beneficial allele would be likely to be dominant. And this might in turn change the way adaptation happens, whether it happens through a hard sweep in which a single copy of an allele sweeps to fixation, or a soft sweep in which multiple independent copies of a given allele sweep to fixation simultaneously. Now, to give you a case study of how this works, I'm going to use the very famous example of adaptation at the ACE locus in Drosophila melanogaster. The ACE locus codes for acetylcholine esterase, which binds and breaks down acetylcholine at the neuromuscular juncture. And because acetylcholine esterase is such an important enzyme, it's often targeted by pesticides, specifically the organophosphate pesticides in this case. And these pesticides bind to the active site of acetylcholine esterase and prevent it from breaking down acetylcholine. Resistance to these pesticides occurs through mutations at the ACE locus, which change the shape of the active site so that pesticides can't bind. Unfortunately, these mutations also make it more difficult for acetylcholine esterase to bind acetylcholine itself. So there is a cost to resistance in this species. Now let's consider what selection looks like before the environmental change, before pesticides are applied. So in this scenario, selection is acting mainly on enzyme efficiency. So there are if you have the 2 wild type copies of the allele, you're doing just great. Your acetylcholine esterase is fantastic. And if you have 2 resistant copies, you suffer some selective costs because your acetylcholine esterase is just not as efficient at processing acetylcholine. And we know based on the metabolic theory of dominance and also based on empirical evidence that the deleterious allele is almost certainly partially or entirely recessive. So if the costs with respect to fitness are recessive. Now, after environmental change, selection has shifted. Selection is now acting primarily on resistance to pesticides, so it's an entirely new regime. Now, if you have 2 copies of the wild type allele, you're in serious trouble because you don't really have functional acetylcholine esterase. Whereas if you have 2 copies of the resistant allele, you're doing really well compared to the wild type homozygote. And again, the question here is what the heterozygote looks like. And we know here that resistance at the ACE locus is semi-dominant to dominant across insect species. And so we know that the beneficial resistant allele is going to be partially dominant. So what we have here is a single resistance allele that went from deleterious and recessive because selection was acting on its loss of function, its loss of binding efficiency, to beneficial and dominant because selection is now acting on a gain of function, a gain of resistance. And we can take classical examples like this with the knowledge we have from these models of dominance and apply them to our current framework. And so we're really interested in how these predicted dominance shifts influence selective sweeps. And to examine this, we turn to simulations. And all the simulations I'm going to show you in this talk were conducted in the evolutionary simulation software. The first thing we had to do was study how selection acts on the deleterious allele prior to the environmental shift. And here, so what we really had to do was look at the mutation selection distribution of the deleterious allele. And we did this across a range of selection coefficients, dominance coefficients, and theta values. Here, theta depends on the effective population size and the mutation rate, but it can be visualized as sort of the mutational supply into the population. Now, what I'm going to show you now is for a particular set of parameters, how the mutation selection distribution changes when dominance decreases. So right now you're looking at the distribution when a deleterious allele is additive. Now I'm going to decrease dominance. And you can see the tail kicking out, the center of the curve sort of comes upward, up to the point when the allele is fully recessive, the tail is very thick and the center of the curve has really shifted upward. Now you might ask, why does the shape of the distribution matter? Why we're not for the mean number of alleles expected in the population under mutation selection? And so I can demonstrate the case. Here we have the same mutation selection distribution, but plotted as a density curve. And we're going to look at what selection, sort of the outcomes after environmental change and after selection. So in situations in which a hard sweep occurred after the environmental change, the number of starting alleles in the population is being drawn from the left-hand side of this distribution. In contrast, in those situations in which a soft sweep occurred after the environmental shift, the starting number of alleles is being drawn from an entirely different part of the distribution, specifically the center of the curve and really extending out into the tail. And this is why the shape of the distribution matters and why dominance changing the shape of the distribution can affect the likelihood of hard versus soft sweeps. So that's one half of the equation. Now, what happens after the environmental change? And essentially what we did is draw from these stationary distributions of different parameter values and simulate many, many different versions of selection. Again, across coefficients, and theta values. For each combination of parameters, we ran 10,000 replicates to really get an idea of sort of what was going on. And we focused exclusively from the standing variation, and I'll explain why. All right, so here's a particular coefficients before. And I'm showing you out of a proportion of 10,000 runs, what the outcome. In this, an allele that was afterwards. This is sort of our control because this is a common assumption made for outcomes. The first is that there was no standing variation in the population when the environmental change occurred. The second is that variation there, but the beneficial allele lost and the sweep failed. The third is that a single beneficial allele went to fixation, which we're calling a hard. Multiple such alleles went to fixation, a soft sweep. So this is sort of our control case, Now I'm going to show you what it looks like when we allow dominance trade-offs before and after the environmental shift. All right, so this is a moderate dominance change from partially recessive to partially dominant. You can already see the pure rate of adaptation from the standing variation has increased, and the relative probability of a soft versus a hard sweep has increased. And as we allow stronger and stronger dominant shifts, this just keeps getting more extreme to the point where it's fully recessive afterwards. And the probability of soft sweep is much more likely relative to the chance of a hard sweep. And we ran, I think, around simulations for over 1 million simulations. So I'm not going to show you all the results, but generally, using these dominant shifts, we can observe soft parameter combinations where you wouldn't normally expect them. It's a case where the selective coefficient after the environment change is acting against the deleterious allele before the environmental change. This is not a scenario in which you usually expect to see a soft sweep. And you can see that by looking on the far left of my screen at the scenario where the allele is additive before and after. However, if we allow some dominance trade-offs, not only does the probability of adaptation from the standing variation increase dramatically, the probability of a soft relative to a hard sweep also increases. So returning to adaptation at the ACE locus, we're sort of lucky in that adaptation at the ACE locus has been studied in terms of hard versus soft selective sweeps. In 2010, a paper was published by Talia Karasov, Philip Mezer, and Dmitry Petrov, which showed conclusively that adaptation at the ACE locus in Drosophila occurred through a soft sweep. However, as the authors point out, given the traditional population size of Drosophila and the resulting theta value of 0.01, adaptation or sweep from the standing variation is unlikely to occur. And what we wanted to do was incorporate what we know about changing dominance in this adaptation at the ACE system into the scenario and see whether that changed anything. So this is a simulation. These are simulation results for what adaptation at the ACE locus could look like. This is a very conservative simulation because I've placed the selective coefficient after at only 0.1. It might and is likely to be much higher. In any case, you can see that when the allele is additive before and after, the probability of adaptation from the standing variation is very low, and the probability of a soft sweep is very low. And as dominance increases, as you allow stronger dominance trade-offs, the raw probability of adaptation from the standing variation increases, and the relative probability of a soft versus a hard sweep increases. If there's a full trade-off, that probability of goes to about 15%. And conditional on a sweep occurring, the probability of a soft sweep is also more likely than that of a hard sweep, depending on the trade-off. Finally, the authors of Karasov et al. Also suggest that the effect of population of Drosophila may have been much larger than the sweep than we thought. And so if we increase theta by an order of magnitude, you can see that, of course, adaptation from the standing variation becomes more likely, and a soft sweep becomes overwhelmingly likely. Really, these likelihood of adaptation from the standing variation and of a soft relative to a hard sweep. We only examine adaptation from the standing variation because these shifts do not change the probability of soft sweeps arising from de novo mutations after environmental change. However, they may play a particularly important role in evolutionary rescue, where adaptation from the standing variation matters a lot. And this is just a case where extraction after some environmental shift can cause the population, or if a beneficial allele is able to fix, it might be rescued. And we find that these increase the probability of rescue versus extinction. These dominant shifts, soft sweeps that derive from these dominant shifts tend to increase the number of alleles and the evenness of their representation in the population. And this is particularly important because this contributes to the detectability of soft sweeps if you can only look at a subsample of the population. And the effect of these shifts is more pronounced when selection before the environmental change is stronger. And this is interesting because one of the geometric predictions of metabolic dominance theory is that more deleterious alleles should be more recessive. So it might not only be that the effect of these shifts is more pronounced when selection before, but that we expect more of a shift when selection before the environmental change is stronger. And finally, these dominant shifts may offer some insight into where in the genome soft sweeps are likely to occur. These dominant shifts can occur in the autosomes, for instance, but wouldn't apply to the sex chromosomes like the X and the Z, which are haploid in males and females respectively. And they apply in diploid insects versus haplodiploid ones. And with that, I'm finished. Thank you so much for listening, and I'd be happy to take any questions.
Okay, thanks very much, Pavitra. Just a quick question. Can your model be adapted to multi-locus interactions? This is from Celine Casey's.
Yes, absolutely. And that's actually hopefully what we're doing next, because we're interested in, as we increase the number of loci, sort of, well, essentially what happens is these dominant shifts become more or less important if you have multiple recessive alleles versus that are trading off into multiple dominant ones. So yes, absolutely.
Excellent. Thank you very much, Pavitra. We need to move on to our final talk for the session. And this is from Cara Brand. She'll be telling us about adaptive evolution at a meiosis gene, mediating species differences in the rate and patterning of recombination.
Thank you so much for the introduction, and thank you for inviting me to present my graduate work during the Crow Symposium. I'm really honored. Recombination is beneficial because at the molecular level, it repairs double-stranded breaks. At the chromosomal level, it ensures the proper segregation of homologous chromosomes. At the population level, it shapes the distribution of genetic variation. And ultimately, it enhances the efficacy of natural selection. Despite these benefits, recombination rates vary across eukaryotes, and this variation occurs both within taxa and between taxa, ranging from fungi and animals and plants. Yet, we're only beginning to understand the genetic factors that cause this variation. Drosophila is an excellent model system to study this variation because recombination varies among species, it varies among individuals, up to 20% in some populations, it varies among the different chromosomes, and even among the different chromosomal regions. For example, crossovers are reduced near centromere and telomere regions and absent in heterochromatic regions. My research focuses on the evolved species differences in recombination rate between the cosmopolitan species Drosophila melanogaster and the island endemic Drosophila marciana, two species that diverged only 2.5 million years ago. Drosophila marciana has a longer genetic map because it achieves more crossovers per each meiosis. Overall, Drosophila marciana crosses over around 1.8-fold more than Drosophila melanogaster, although this rate does vary among the 3 major chromosomes. The chromosomal distribution of crossovers also differs between the 2 species. To demonstrate this, we plot the physical position of a locus on the x-axis against its genetic position on the y-axis, and most importantly, the slope of the line provides information about the rate of crossing over across the chromosome. In Drosophila melanogaster, this slope is nearly 0 when it's close to the centromere, and this indicates that crossovers are essentially absent in this region. In contrast, the positive slope in Drosophila marciana indicates that crossovers do form near the centromere, demonstrating just one example of how crossovers are patterned differently between the species. My research investigated the underlying genetic factors responsible for the evolved species differences in crossover rate and patterning between Drosophila melanogaster and Drosophila marciana. And I began this work by taking a candidate gene approach and focused on a meiosis-specific gene called MY217, MY218. And this gene is peculiar because it's dicistronic, which means that it's transcribed as one long transcript with 2 translation start sites that give rise to 2 separate proteins, MY217 and MY218. And both MY217 and MY218 are part of a meiosis-specific complex that's required for crossover formation. As a result, nearly all crossovers are eliminated when either protein is knocked out. The MY218 protein was particularly compelling because it displays unusually high protein coding divergence. In fact, the DN-DS ratio of MY218 falls within the 97th percentile of all genes in the genome. To investigate the evolutionary forces causing this protein divergence, we performed population genetic analyses that uncovered a strong signal of positive selection across MY218. As a result, between Drosophila melanogaster and Drosophila marciana, there are over 200 fixed non-synonymous substitutions across MY218, and these sites comprise over 12% of the coding region. To investigate if evolution at MY218 plays a role in the evolved species differences in recombination rate, we took a transgenic approach and compared recombination rates between females with nearly identical genotypes. On the X chromosome, the endogenous copy of MY218 is knocked out. We then inserted either a melanogaster or a marciana allele of MY218 as a transgene on the 3rd chromosome. And finally, we measured crossover frequencies between visible markers that span the left arm of the 2nd chromosome and then also span the centromere. And while we expected that the melanogaster MY218 transgene would complement the mutant endogenous gene and rescue recombination rates, it was unclear how the Marciana transgene would behave. We envisioned 3 possible scenarios. First, the Marciana transgene may be too diverged to function in a melanogaster background and fail to rescue recombination rates. Second, the Marciana transgene may actually rescue recombination rates, but do so at melanogaster-like levels. And this would indicate that the divergence that we observe at MY218 is not responsible for species differences in crossover rate and patterning. And last, and perhaps most interesting, the Marciana transgene may rescue recombination rates and produce elevated Marciana-like levels of crossing over. Unsurprisingly, the melanogaster MY218 transgene rescued the mutant phenotype and produced wild-type crossover levels across the region. The Marciana MY218 transgene also rescued recombination rates, but did so at elevated levels that surpassed the melanogaster-like levels. In total, the Marciana MY218 transgene recapitulated 82% of the wild-type Drosophila marciana genetic map. This outcome was striking to us, that a single gene would have such a large effect on a complex genome-wide phenotype like recombination. However, when we look across the individual genetic regions that we measured, we notice that this rate increase is heterogeneous. And this suggests that the Marciana MY218 transgene may also influence crossover patterning. Crossover patterning occurs because crossovers are regulated in 3 major ways. Crossover suppression mechanisms prevent crossovers from forming near centromere and telomere regions. Crossover assurance mechanisms ensure that 1 obligate crossover forms per chromosome. And crossover interference mechanisms prevent 2 or more crossovers from forming near one another. Beginning with crossover suppression, we find that significant crossover rate increases only occur in the telomere proximal, centromere proximal, and centromere spanning regions, indicating that the Marciana MY218 allele yields weaker crossover suppression. To measure crossover assurance, we calculated the estimated number of crossovers per each chromosomal tetrad. While the melanogaster and Marciana MY218 allele produce approximately the same amount of single crossover chromosomes, the Marciana allele produces fewer non-crossover chromosomes. And this indicates that the Marciana MY218 allele yields stronger crossover assurance. The Marciana allele also produces more multi-crossover chromosomes, suggesting that crossover interference may also differ. To quantify this, we calculated the strength of interference between the 2 largest adjacent regions and found that the Marciana MY218 allele yields 36% weaker interference. So to summarize this section, our research found that the Drosophila marciana MY218 allele causes weaker crossover suppression, stronger crossover assurance, weaker crossover interference, and together these forces contribute to an overall elevated crossover rate. Ultimately, the 2 MY218 alleles produce different crossover landscapes, both of which may influence chromosome segregation. For example, weak crossover assurance results in chromosomes that lack crossovers, and these chromosomes are at risk for non-disjunction events during meiosis that lead to deleterious aneuploid gametes. On the other hand, weak crossover interference results in chromosomes with multiple crossovers, and these chromosomes are also at risk for non-disjunction. To test if either the melanogaster or Marciana MY218 allele has repercussions on chromosome segregation, we measured non-disjunction rates of the X chromosome and found that the melanogaster MY218 allele yields 2-fold more non-disjunction events. And this suggests that the Drosophila melanogaster crossover strategy is unnecessarily vulnerable to costly non-disjunction events. What evolutionary forces could outweigh this cost of non-disjunction and shape crossover landscapes? Additionally, what forces can drive recurrent bouts of adaptive evolution? And we speculate that selfish genetic elements such as transposons may be the evolutionary force driving crossover rate differences between the species. Transposons pose a significant threat to recombination because non-homologous exchange between similar but dispersed transposon sequences can generate deleterious chromosomal aberrations. While we often associate transposons with heterochromatic regions of the genome, many copies of transposons are found within euchromatic regions. For example, this is just a snapshot of the numerous transposon insertions across the left arm of the 2nd chromosome in Drosophila. Additionally, a previous study found evidence for transposon-mediated duplications and deletions in over 2% of meioses, which is a substantial detriment to fecundity and is likely a significant selective pressure on recombination. The rate of non-homologous exchange also fluctuates over time, tracking with the fluctuating load of transposable elements. When a new transposon family first invades the genome, the risk of ectopic exchange is low. But this risk increases as transposons proliferate across the genome. Over time, transposon copies will diverge and the risk of ectopic exchange will begin to decrease, and eventually become very low as the transposon copies decay from the genome. However, this process is continuous as new transposons invade the genome. Most importantly, this process occurs independently among the species. Therefore, the risk of non-homologous recombination will differ depending on the species-specific transposon load. Under this model, the evolutionary causes of species differences in crossing over emerges as species evolve towards an optimal recombination rate that balances the cost of non-disjunction against the risk of ectopic exchange. And this process fluctuates over time due to recurrent transposon invasions. And while it's empirically hard to test this model, we can, however, make observations about the downstream evolutionary consequences of species differences in recombination rate. So we heard in this session about how recombination rate can affect introgression in gene flow, but there's also another well-known observation about how nucleotide diversity is correlated with recombination rates. Recurrent positive and negative selection reduce nucleotide variation at genetically linked sites. These so-called hitchhiking effects are ameliorated by recombination, giving rise to genome-wide correlations between nucleotide variation and recombination rate. We see this pattern in Drosophila melanogaster when we plot nucleotide diversity across the second chromosome and notice this valley of reduced diversity in the region surrounding the centromere. This same pattern also emerges for Drosophila marciana. However, when we compare these two species, we notice that nucleotide diversity is overall higher in Drosophila marciana. And this is unusual because nucleotide diversity is directly related to population size. At first glance, this seems unusual that the island endemic of Drosophila marciana would maintain overall more nucleotide diversity when it certainly has a smaller census population than the cosmopolitan Drosophila marciana. However, adaptive evolution at MI218 contributes to this difference because, as I've already shown you, it mediates the strength of crossover suppression. In Drosophila melanogaster, crossover suppression mechanisms are strong. Therefore, this domain of reduced crossing over extends further from the centromere compared to Drosophila marciana. As a result, levels of nucleotide variation recover less quickly in Drosophila melanogaster than in Drosophila marciana. So to close with the summary of my research, we found that adaptive evolution at MI218 is directly responsible for species differences in crossover rate and patterning, and it modulates recombination through crossover suppression, assurance, and interference mechanisms. The rate of crossing over also influences genome-wide levels of nucleotide variation. Therefore, adaptive evolution at MI218 incidentally shapes species differences in the chromosomal distribution of nucleotide diversity. And finally, we speculate that these phenomena are driven by selfish transposable elements. And before I end, I would just like to quickly thank the Pressgraves Lab at the University of Rochester, where I conducted this research, particularly my PI, David Pressgraves. And I would also like to thank my current lab, the Levine Lab at UPenn, where I'm currently a postdoc, and I will take any questions if there's remaining time.
Thank you very much, Cara. We do have time for a question. This one comes from Abby Dernberg. She says, great talk, Cara. A possible explanation for the increased crossovers and lower apparent interference seen in Mauritiana is that there are more class 2 or non-interfering crossovers. Have you or anyone examined this, for example, by counting recombination nodules?
I have not done that. What we're trying to do, I started this project as I left the lab, is do a fine scale recombination map in Drosophila mauritiana that uses next-gen sequencing to try to tease out gene conversion events, which I haven't talked about, but then these class 1 versus class 2 recombination events. So we could really better correlate all the dynamics that are going on, but particularly correlate how recombination rate specifically affects nucleotide diversity and other population genetic parameters across the genome.
Okay, thank you very much, Cara.
Thank you.
And I'd like to thank all 5 of our outstanding speakers today for truly exciting talks. So send your applause their way. I'm sure they can feel it. And I'd just like to remind you that you can tune in on Saturday in the Future Visions of Population Evolutionary and Quantitative Genetics session to hear the announcement of the award and also to hear exciting talks from our Crow Award selection panelists. Thank you very much for attending, and we'll see you later.